Professor, I had a question.
Oh, what's up?
With like the extra credit, like when we do that, like I did two different
Are
ones.
you
And, like,
the one who's tried to email me about it?
yeah, yeah, I did. Yes.
Okay. I apologize. I was trying to figure out how to explain this, and then I forget where your email was and when it came.
Okay.
I was actually thinking about it earlier today. I have to put them on. I have to post them there on Gradebook, or they don't get accounted into the grades. And that also means that they're going to up the number of points. However, when I go to add up the points at the end of the semester, there's only certain points that are things that were required.
Dropped off therapy earlier.
And those aren't required. And that's why they're not going to count towards the required grades.
Okay, so like even though it adds it to what the total is, like
Right.
you're saying when you do it at the end, you don't do it that way.
No, and I'll take they'll be removed. So for example, if you right now they're at the end of the semester, and I'm trying to remember, I think it's 1,250 points approximately.
Okay.
If we get all of those assignments done, which I'm not sure we will, but if we get all of them done, if you did all of the extra credit, it's going to give you a total of, if you got 100% on everything, it would give you a total of 310 points. Excuse me, 1,310 points. I knew that didn't sound right.
Okay.
1,310 points.
And it would be out of the
1,250.
1250. Okay.
Does that help? Does that explain?
Yeah. So I just, because like when I was looking at the grading and it said like how many points it was as and what mine was, and then I would like calculate my percentage like for my grade, like I noticed it didn't change because both my points and the total points went up. And so that's why I was confused, because I'm like, I feel like I'm doing it for nothing then, because it's just keeping my grade the same because it's adding it to both. So
Right.
yeah.
Yeah, it's adding them because it's telling you just how many points you have altogether for the class.
Okay.
Does that help?
Yeah, I think so.
Okay.
Thank you.
So I mean, it's going to yeah, in the end, it's only going to be out of 1,250 points. So anything extra beyond that is going to help your grade regardless.
Okay, so basically what I'm looking at in the grading isn't 100% correct if I did extra credit, is what you're saying. Like,
it's giving you your total number of points, but it's not the total number of points that you've worked towards is not the required total.
okay.
Your required total is less than that.
Okay. Thank you.
Okay. You're welcome. Other questions or concerns? She was asking about the extra credit and that it's automatically upping up your total points whenever you do extra credit. But the total for the semester is 1,250 points. If you did all of the extra credit and you got 100% on everything, your total for the end of the semester would be 1,310 points. But then you take that 1,310 points and divide it by the 1,250. And that's really what your grade is. So it would be like 105% or 100 or 3% or something like that. Does that make sense to everybody? And so it's adding your points every time you do those. But I'm going to take into account that whatever points that you earned from those extra credit assignments is added onto what is separate from the required points and the required assignments. Let's jump into tonight, unless somebody has something else to bring up.
When it is due lab quest number, let me see.
I'm not sure. I can look on McGraw-Hill and tell you
No, I'm sorry. Case story.
the case study.
Yeah.
The cranial, so the first one was due a while ago. If you haven't done it, just submit it.
No, I did that one.
The cranial nerves.
Yeah.
Okay. The cranial nerves case study would be due most likely next Sunday or the following Sunday.
Okay.
So what I would, and the reason, let's say the following Sunday, so 2 weeks from yesterday. And if you look at it, and I was going to jump into that first anyway, so let's talk about that right now. If you look at this one, it's different than your first case study. It's set up a little differently. I think in a lot of ways it's easier, and in some ways it's harder. But I still think it's easier overall.
I have no score for the first one, so I don't know how that works.
What?
I do not have it's a score.
I haven't graded it yet. That's why you don't have a score yet.
Okay.
I will get to it.
Okay. Just want to make sure.
But let me show you what the second one here. So what it is it's not cranial nerve case study 3, it's case study 2. But describe a clinical presentation case scenario when assessment of cranial nerves is warranted. You can really put almost anything here. For example, someone has a stroke. Someone gets hit on the head. Someone sustains some sort of head trauma. All of those would be reasons for a clinical study and assessment of the cranial nerves. Name at least 4 muscles controlled by the oculomotor nerve. We talked about this in lab. We'll go over it again a little bit tonight. But if you go back and you look at lab, you will find all 4 muscles. Besides eye muscle innervation, what other functions does cranial nerve number 3 have? Go back and look at lab again. At the lab, and you'll find the answer. This one, case study, a 61-year-old female with a history of an acoustic neuroma, which you don't honestly need to understand, but acoustic means sound, neuroma meaning a tumor, with right-sided facial paralysis and the inability to close her eye, which cranial nerve is most likely affected. We will talk about it tonight. You'll see it tonight. The temporalis, medial lateral pterygoids, and masseter muscles are innervated by which of the following? We saw it in lab, but you can also Google this. If you say, what cranial nerve controls temporalis, medial lateral pterygoid, and that P is silent, so it's pterygoid, and masseter muscles, it will tell you what cranial nerve is involved. The facial nerve innervates which of the following? You guys you can maybe figure this one out from the first one. Which reflexes involve optic and oculomotor nerves? This is another one that you could Google if you wanted. Describing a pupillary reflex and a consensual reflex and an accommodation reflex. If you go to your textbook, they're all in there. Or you can also Google the answer. What are 3 sensory branches of the trigeminal nerve? We talked about them in lab, but you can also find them if you go online. Male, so if you get the general idea, so between lab lecture and Googling things that you're not sure about, if you get it on Google, do me a favor and provide a citation. Because if it's not the answer I'm expecting, I'm going to wonder where you got it. And altogether, there's 27 of these. If you remember this last one, which cranial nerves carry only sensory, which carry motor, only motor, which ones carry sensory and motor? Remember that some say money matters, but my brother says blah, 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 blah. That's the answer right there. Don't write down that mnemonic, but what cranial nerves are only sensory, what ones are only motor, and what ones are both? Do you see why I think it might be easier? How would you test the cranial nerve number 9? Don't put on here the one of the answers that people like to give, and I'm not sure why they give this, is how can the cranial nerve be tested? We'll test the muscles that the cranial nerve innervates. You guys, that's not, of course, that's the way you do it. But I want to know, how would you test those muscles? Not that you test muscles that nerve innervates, because that's already kind of that's a given in my book. Go and look, and you can look this up. How would cranial nerve number 11 be tested? And it will tell you what muscles are involved. Any questions about this?
How detailed is that clinical presentation that we're supposed to create be?
It doesn't need to be that involved. Someone sustains a head trauma. Someone has a patient comes in who's had a stroke. That's all I want.
Okay, thank you.
The one thing that I don't want is describe a clinical presentation when an assessment of the cranial nerves is warranted. Don't put something like what's an answer I've gotten in the past that makes me crazy. When you think damage to the cranial nerves can be suspected. Well, what would cause potential damage to the cranial nerves? That's what I want you to tell me. Don't say because we'd assess them if we thought they were damaged. I know that. But I want to know what kind of injury would be involved. A stroke, a head trauma, a more nastier adventure. Someone undergoes a time of has temporary hypoxia, and you want to make sure that their cranial nerves are still intact and functioning. All those would be possible answers. Does that help, Christina? Okay.
Yes, thank you.
All right. All right. So let's jump into tonight. And my goal tonight, I'm going to try to finish the majority of chapter 14. And if you'd be willing to turn on your cameras, I'd appreciate it. But I also understand.
I was just curious, has anyone else taken a long time to do lab 20? Because I spent like 2 hours and I only got like 60% done.
What one was 2020 was the brain.
I've
Yeah.
only started the lab, but chapter 12, like everything lately seems to be taking 152 years. I almost ran out of time on my chapter 12 quiz.
See, you said the same thing about chapter 2. I don't believe you now, Justin. I don't believe you.
I'm telling you, some things I can zoom through, but 12, man, really tripped me up.
Are there specific questions that are harder than others, Christina?
Yeah, some of the diagrams I feel like we didn't have in our book or our PowerPoint or even the lab book. And so the really detailed areas, I feel like, especially, you know, certain views, I just felt like I struggled a whole lot and it took me a long time
Let me
to look
see.
in, and
Not
I'd
that
have
one.
to redo, and after like 3 attempts, I just used the correct answer because I was like done. I mean, I
Is-
got a lot of them right. They just took a long time and a lot of like looking back and forth because everything is so close together.
Let me look just for a quick second.
Like once I got through like the first 20 questions, it wasn't so bad, but the first 20, you know, would have like 3 diagrams in one question. I'd be like, no.
Yeah. Look. Let me see.
I feel like I'm really struggling with like the different parts of the like gyruses, you know,
Different
and
parts of the gyruses.
like where the gyrus and the speculus and like those little smaller
The
parts,
what? Say it one more time.
like identifying if something is a gyrus or if it's just a lobe, because sometimes those diagrams are like asking for more detailed things or bigger, and it's hard to tell, like, is it just saying it's the frontal lobe or is it saying it's, you know,
Can you give me an example as I'm searching for something?
Sure, let me see if I can look to see what question. I took some screenshots. Well, like question 21, I struggled with that one. It was a diagram.
It not what am I on 21? No, I'm on 14, Mary. That's why I don't see it. Well, areas of the cerebral cortex.
Because I don't
Go,
know, whatever I
go.
was doing to help me was slightly different, and so
What does it give you as? Of course, it won't let me scan them. What does it? Here, let me do something a little different. I don't know. I don't want this. Yeah, let me see if my student view gives me a different thing than what you have.
It's not lab exercise 20 that's due tonight, is it?
No, it's due tomorrow night. Student view.
I thought it said do the 8th or no, the 6th.
So you know what, Christine, I can't can't hear you.
I think it's due the 6th.
Yeah. Yeah, that's fine too.
But be careful with that because when you look at some of the great, I think one of them was showing like 12 a.m. on the 6th. So I think it's always, remember that it's always the Tuesday that it's due, right?
Yes,
Uh,
it should
okay.
be. So one, where's one? Okay, that looks like Broca's area. Where did we say that Broca's area was? Our, bear with me as I sometimes growl at my screen. Of course, it's not going to work. Really? Let's try one more time. Let's just see what happens. I'm going to try one more time and then I'm going to try going to a different class. You guys, I just opened it 2 minutes ago. I actually opened it right before class and it was opening fine. Let's see here. Actually, let me, before I spend a lot of time with this, because we will be looking at these regions of the brain again.
Yeah, you can talk about it later. I was just mentioning that some I felt were more difficult than others.
Do me a favor. Let's do this. If you're able, and I don't know how much computer technology you have, like I have 2 computers. I know I can't access things on my phone, but I know other people can. If you can find the questions that you think you're going to struggle with the most, or ones that you know you're going to struggle with more than others. And when we come to those, most of this we're going to address again tonight. When we get to those, answer those questions as we go. Is this clear what I'm saying?
Yes, thank you.
Okay. Let's see really quick if I can open this up again, because I swear I just had it open right before class. All right, let me see if I can open it through another course, but it's the same PowerPoint. I think it's the same PowerPoint. We'll find out. So I think I was trying to tell someone that they should not take 122 during the summer. I don't remember who that was with, or Hack does it differently. And I know this because I just got asked to teach a 121 course over the summer. There are classes that are there's 121 and 122 that are 3-month classes. And there's 121 and 122 courses that are 5 months, assuming not 5 months, 5 weeks. If I were you, if you feel really confident with AMP already, then take 122 over that 5-week, 6-week time. But if you still feel like you're maybe not as confident, because as Zena can probably tell you, we cover a lot in 122. If you feel like you're maybe not as confident, Hack also offers you a chance to take it over the whole entire summer. I think it starts in May and it ends in August. And so that's another option for taking 122 if you think you need to take it over the summer. And I said that because I said something about it earlier, and Hack's the only school that I know of that offers that kind of thing. Or 15, 8. There. Maybe. Okay, so we just couldn't open it on our course, but this is the same exact PowerPoint. And this is just one from last semester. It's a course site from last semester, but your PowerPoint is the same one. So we were on 17. So the two places, so talking about arterial blood supply of the brain. The brain receives blood from the two pairs, the pair of internal carotid arteries and the basilar arteries that's formed from, the basilar artery is formed from the fusion of the two vertebral arteries. And then it comes up, here's the vertebral arteries, they fuse to form that basilar artery. You have a posterior cerebral arteries, middle cerebral arteries, and anterior cerebral arteries, starting anterior, posterior. Internal carotid arteries coming up from it. We're looking at a inferior view, so these are going to be coming out at us here. The internal carotid arteries and the vertebral arteries coming up via the transverse foramina and the cervical vertebrae. When the blood gets drained from the cerebral area, it's going to drain via the dural sinuses that are going to empty into the internal jugular veins. A superior sagittal sinus, the inferior sagittal sinus, the straight sinus coming down below, coming down along the tentorium cerebelli, and the inferior edge of the fossa cerebri. Remember that the other thing that this is important for is that, and we'll see it again in a little bit, is that this is where the cerebrospinal fluid gets returned to the bloodstream. It gets created from the blood, it gets returned to the blood via these sinuses. The blood-brain barrier vascular endothelium around the brain forms tight junctions with the perivascular feet of the astrocytes. What were astrocytes?
A type of nerve cell?
You're close. What can you be more specific? Because there's two general types of nerve cells. Are they neurons or are they, what else could they be?
Glial?
Yep, they're the neuroglia cells. So these tight junctions, if you remember these from our histology chapter that we had in lab, these tight junctions are going to prevent things from moving through the cells. Excuse me, prevent things from moving in between the cells and forcing them to move through the cells. So tight junctions that are going to prevent things from being able to move in between the cells and therefore make them move through the cells. These tight barriers are going to ensure that only essential metabolites are going to be able to move into the capillaries of the brain, but prevent the passage of larger, more hydrophilic things like red blood cells from moving from the blood into the cerebrospinal fluid. It's going to prevent things that are hydrophilic, generally large hydrophilic things. It's going to prevent them from being able to move into the red blood, into the blood circulation of the cerebrum and cerebellum. So it's strictly going to regulate what can get from the bloodstream into the tissues. It's an obstacle for delivering medications such as antibiotics and cancer drugs. If you remember, I talked about my cousin and the glioma that he had removed almost, I don't know, about a year and a half, 2 years ago. The only real way of treating most neurogliomas, or AKA neural tumors, is removal, because chemotherapy drugs don't work because of the blood-brain barrier. Trauma and inflammation can damage the blood-brain barrier and allow pathogens to enter the brain tissue. There are certain areas, circumventricular organs, and just from that name alone, you can maybe guess where these are. Circumventricular, meaning they're going to be found within the ventricles. And remember that you only have 4 ventricles. These are found within the 3rd and 4th ventricles, where the barrier is absent. And so this is a place where the blood has direct access to the brain. And it allows for the brain to monitor and respond to fluctuations in blood glucose, pH, osmolarity, and other variables. One thing that we learn later, like next semester, and also a little bit tonight, is that the hypothalamus is a major part of monitoring fluctuations in blood glucose kind of, but more pH osmolarity. Well, this is how it can do that. It can monitor because of areas where the blood-brain barrier is absent. Nevertheless, the circumventricular organs allow for routes of invasion, for example, by the human immunodeficiency virus or HIV. Why is the blood-brain barrier ineffective against alcohol? And of course, that's a typo. Why is it ineffective?
Is it D?
It,
As in,
you know what, and I don't really have an answer to this. This isn't a question that would be asked on an exam because I don't really have an answer to it. But I would say that first off, we know that the blood-brain barrier is preventing against things that are large and hydrophilic. So if it's lipid soluble, that would allow it to easily pass through the blood-brain barrier. So it's not D as in Delta. It can reach the brain, so it's not C either. I would say we know that the blood-brain barrier prevents against blood cells, but blood cells are a lot bigger than alcohol is. I would say that it's because alcohol is water soluble. But, and it's actually, let's split this one. It's water soluble, but it's also really, really small. So it's actually, the answer would be B as in Bravo. Because it's only gonna be, it's not gonna be so much as just red blood cells. It's gonna be larger things. So the reason it's ineffective is it's only working against larger molecules, not little tiny hydrophilic molecules. Alcohol is almost the same size as water itself.
And I found an interesting answer on this one. It says it's also due to its chemical characteristics, although it's somewhat polar, it's also lipophilic.
Yeah,
And so
it's both.
so it mixes easily with fat in the membrane.
Yeah, it's both. It's got, bear with me, I'll show you because I was just looking at it earlier for another class I have. It looks like this. So it's got a CH3. So it's got one methyl group, and that's not what I meant. It's got another CH2. And then it's got a hydroxyl group. This is the typical chemical structure of ethanol, which is what you find in alcoholic beverages. Like Justin just said, this structure right here is hydrophobic, but this part is hydrophilic. So it's one of those amphoteric molecules or amphiphilic, where it's going to be in both places. It's more soluble in water than it probably is in lipids because it's so small. An alcohol that had more CH3 or CH2 on it is going to be much more lipid soluble. But the fact that it's small means that it can be hydrophilic and also slightly hydrophobic too. As far as I know, if you put rubbing alcohol, which is very similar in structure, if you put that into an oil, it doesn't easily dissolve. Isopropyl alcohol. And isopropyl alcohol is much more hydrophobic, meaning it should dissolve in lipids a lot easier than this does. I mean, you can try to do it. I would be curious what would happen because I've never tried it, but try putting some of your rubbing alcohol, or actually maybe not rubbing alcohol. try putting some of an alcoholic beverage in water, oil, and see what happens. I don't know, actually it might be a very good experience because most alcoholic beverages are mainly water anyway. But I don't think it's that hydrophobic. I don't think it's that lipid soluble. Does that make sense? It's more water soluble than it is lipid soluble. I think if I really wanted to, because I can really, it's amazing what you can buy online. You've seen how many fixed organs I can find. If I really wanted to, you can go on eBay and buy 100% ethanol. But I won't do that in the time that we have class left in this semester. That will wait for another day of experiments. My mother likes all the experiments I put around the house anyway, as she laughs in the background. Okay, all right. So I think that it is most likely. Okay, someone. Okay, Jennifer, I understand. I hope your eyes are doing all right.
Thank you.
You're welcome. So I think it's mainly that it really only prevents larger hydrophilic molecules from not being able to pass. That's more likely to be the right answer. So B is in Bravo. Which of the following is not a true statement regarding the blood-brain barrier?
Is it E?
B is in Bravo.
No, E prevents the flow of, because we're saying what's false, right?
Yeah, what's not true? Well, does it contain gap junctions?
Yes.
Are you sure? Are they gap junctions or are they tight junctions?
Okay, you got me on the junctions.
You're all right. You're all right. You know, I'm the one who's really familiar with the words, so you're all right. But yes, it's going to prevent the flow of large protein. Well, you know, really any protein, or let's say more hydrophilic proteins, because like Justin said, hydrophobic proteins are not really going to be prevented. Ventricles are cavities within the brain. The brain has the consistency of cold cooked oatmeal. You kind of saw what it looked like the other day. I did throw that away that night. I should have maybe saved it for another lab in the future, but we didn't. The ventricles of the brain are filled with cerebrospinal fluid, and they essentially keep the brain from collapsing in on itself. I don't know if it really has exactly that texture, because freezing it is going to affect the texture. And the fact that the second you kill something like a pig, it's going to start deteriorating until you freeze it. So whether it had the exact same consistency as what we saw last week, I can't tell you. But I would think that it would be fairly close. The cerebrospinal fluid circulates through the ventricular system. Remember, it's secreted in the lateral ventricles, the third ventricle, and the fourth ventricle. Anywhere that's lined with ependymal cells is going to secrete cerebrospinal fluid. So cerebrospinal fluid continually circulates through the ventricles and spinal cord. It's constantly bathing the brain and spinal cord with oxygen, nutrients, and vital chemicals. Its main functions mechanically, like Justin made a comment about in lab last week, the brain is essentially in the spinal cord are essentially floating in cerebrospinal fluid. If you've heard of people who have tried to get up after a lumbar puncture, or as I had a student a couple semesters ago who actually had leaks of cerebrospinal fluid, and they finally found out why she was having severe migraines regularly, was because she was having leakage of her cerebrospinal fluid. I don't know how she would have just did that over and over again and not felt like she was going to die. But your brain and your spinal cord are essentially floating in cerebrospinal fluid. Without the cerebrospinal fluid to support it, portions of the brain would sink through the foramen magnum. It also, because of the flotation of it and the fact that the brain floats in it, it's going to provide a liquid cushion that protects delicate neural structures from sudden movements. Like trying to walk quickly in a swimming pool. The water is acting as a movement buffer. Homeostatic functions. It transports nutrients and chemical messengers to the brain and removes waste products from the brain. It's going to protect nervous tissue from chemical fluctuations that would disrupt neuronal function, like osmolarity, pH. Osmolarity, we haven't really talked about osmolarity before. Think of molarity, which you probably haven't thought of before either, but I was just explaining to my physiology students at Stevenson, think of osmolarity as a concentration.
I actually had a patient today that was getting checked for osmolarity because he
Wow.
was excreting too much urine. Like he literally filled a hat. One of the hats, the toilet hats.
Oh, okay.
With-
I'm like, he filled the hat? What was he? Never mind. Okay.
Yeah, he filled the hat, and like we had to send his urine out and check for osmolarity and blood serum osmolarity because he was excreting like over the threshold of urine.
Over the renal threshold or
Yeah,
transport maximum.
because
Okay.
I actually asked the nurse practitioner I worked with. I was like, "What's the osmolarity? What does that check for?" And he was explaining it to me.
What did he tell you?
He said something about like, well, because then I had to do a urine dipstick, a urinalysis, and I was
Yep.
like, he had glucose in his urine, and he was like, so when your like diabetes is uncontrolled, or if you have osmolarity issues, it can start excreting in the urine.
Right.
And then he was saying like something about like your renal threshold is only able to handle 150 or 180 something.
Yep.
And he was like, any more of that can cause like osmolarity.
I think it's
like,
higher than that, but according to the textbook, it's higher than that. It's about
something
300.
like that. It was really early in the morning. He was explaining it, so
I understand.
Think I can remember some of it right now.
Well,
and I know he sent him out to get an MRI or CT of the brain or something. He had to go get done today, like a stat order.
Well, a really easy way to think of osmolarity is concentration. So if you think of a juice that you make from a concentrate, what are you doing when you make juice from a concentrate? You add a bunch of water to it, right? So when your osmolarity of your blood goes up, what it's essentially doing is it's saying that your body is low on water. And it's more concentrated than it should be. But yeah, that's awesome. See, that makes sense to me. And next semester, when you learn about 120, when you're in 122, or this summer, or whenever you decide you're going to take it, bring this up again, Sam. So cerebrospinal fluid is going to protect nervous tissue from chemical fluctuations that would disrupt neuronal function. Waste products and excess cerebrospinal fluid are eventually transported into the venous circulation. Remember, how do they get there? They're getting there through the sinuses. They're getting there through the subarachnoid villi that are in the subarachnoid space up here. So the cerebrospinal fluid is produced by the choroid plexus. Just as a sneak peek about what's going to happen Wednesday night, we're going to see a choroid again there. Remember, what did I tell you the choroid plexus looked like? We saw it a little bit in our sheep brain the other night. What did it look like? What color was it?
Like black.
It's black because it looks like dried blood when it's fixed. So just like we're going to see a choroid layer when we look at that eyeball that Ivy really wanted to look at, and that's the whole reason I got it. It's going to look black also. So it's an area that's just coated in tiny capillaries that are lined with ependymal cells. And as the fluid leaves the blood, it moves through the ependymal cells and gets filtered out of the ependymal cells as cerebrospinal fluid. Justin, I just saw your dog lick you. That was really funny.
She is bothering me. She wants to go outside, which is always when I'm doing something.
Okay, don't say anything like W-A-L-K because
Exactly.
I have one of those too. Don't do that. So cerebrospinal fluid flows continuously due to its own pressure gradient. Things are always going to move from from a higher pressure to a lower pressure. And the beating of the cilia, can't see it really well, the beating of the cilia on the ependymal cells and the rhythmic pulsations of the brain due to the heartbeat are going to allow for the flow. So looking at the circulation of the cerebrospinal fluid, it's going to be secreted in anywhere that's lined with ependymal cells. Going to be secreted in the lateral ventricles, the interventricular foramen into the third ventricle, which is going to move, the cerebrospinal fluid is then going to move via the cerebral aqueduct into the fourth ventricle. As far as I know, all of these are lined with ependymal cells. I have never Googled whether the cerebral aqueduct is lined with ependymal cells, but I would think that it would be. It's going to move from the cerebral aqueduct into the fourth ventricle and then into two lateral apertures, openings, and a median aperture into the subarachnoid space and the spinal cord. We don't have the spinal cord on here, but the spinal cord move into the subarachnoid space and return to the blood via the subarachnoid villi to the superior sagittal sinus. So this is showing that same idea with a picture. So it's created within from the choroid plexus. It's going to flow through the interventricular foramina into the third ventricle. Right here. Here's the lateral ventricles. Here's the third ventricle. The choroid, and then the choroid plexus and the third ventricle is going to add more cerebrospinal fluid. And the cerebrospinal fluid is going to flow down the cerebral aqueduct into the fourth ventricle, where the choroid plexus is going to add even more cerebrospinal fluid. It's going to flow in out of the two lateral apertures and the one median aperture, where it's going to fill the subarachnoid space and the brain and the spinal cord. And at the arachnoid villi, it will be reabsorbed. Memorize this pathway. Hydrocephalus is an abnormal accumulation of cerebrospinal fluid in the brain. What does hydrocephalus mean?
It's like swelling of the brain, right?
Well, you're close. You're right. You're right,
Like
but
fluid in the brain,
what does hydro mean?
water.
Water, cephalus meaning brain. So this is essentially water on the brain. So like Christina said, it's a swelling of the brain due to abnormal accumulation of cerebrospinal fluid. This is where they have to go in and put in a shunt many times to help with a blockage in the flow or reabsorption. It can occur with congenital abnormalities, a head injury, meningitis, episodes of bleeding into the brain, and tumors. A tumor that blocks somewhere in this route by its enlargement is going to cause swelling, right? If it can't be returned to via these arachnoid villi, it's going to just occur and swell. So in a baby who's fontanelles, remember what those are? Those are the little places where the skull plates have, cranial plates are not fused. The head bulges due to the increased pressure. The brain stem, you guys, any questions, thoughts? The brainstem is continuous with the spinal cord. It's divided into three parts: the midbrain, which includes the cerebral peduncles. It's a great word, peduncles. Peduncle meaning feet. The pons, and the medulla oblongata, which includes the pyramids and it's connected to the spinal cord. From which vesicle does the medulla oblongata develop? So these were those 5 ventricles.
The A,
It's a good job, Sherry. I was gonna say, where would you find it? And she found it all automatically. The medulla oblongata includes a pair of pyramids, which are pairs of ridges separated by an anterior median fissure. 4 pairs of cranial nerves are going to begin or end in the medulla. Cranial nerve number 8, cranial nerve number 9, 10, and 12. So remember a mnemonic for the cranial nerves. I'm going to do the one that's more cleaner because first off, my mother's here now. But she didn't hear that. I'm sure she did. On they traveled and found Voldemort, cranial nerve number 8, vestibular cochlear. And on they found Voldemort guarding glossopharyngeal, very vagus, ancient horcruxes, hypoglossal nerve. Pyramids are also going to contain the descending tracts or descending fibers of the corticospinal tracts, which carry motor signals to skeletal muscles, voluntary motor signals. All ascending and descending fibers connecting the brain and the spinal cord pass through the medulla oblongata. Also within the pyramids, we have a decussation. What does it mean, decussation? What does that mean? I'm going to slide over some of this. What does decussation mean?
They cross over.
They cross over. So axons from the left pyramid are going to cross over to the right pyramid, and axons from the right pyramid are going to cross over to the left. The result is that the left hemisphere of the brain controls the voluntary movement. Remember, corticospinal tracts, voluntary spinal movement. The left hemisphere of the brain is going to control voluntary movement on the right side of the brain, and the right hemisphere is going to control voluntary movement on the left. You guys, another thing that I should mention is that if we finish this tonight, which I don't know, we'll see what happens. If we finish this tonight, your second exam will be posted sometime within the next day or two. I can't say it's going to happen tonight, but it will be within the next day or two. It might be tonight. Let's just see. I can't remember how much headway I've already made on it.
How long would we have to do the exam?
You will have, how much time did you think you had for the last one? You remember, Christina?
I feel like you said it was like 80 seconds per question.
There's a lot of questions.
But I was also asking, like, do we have like a week to?
Okay,
Yeah.
that's okay. See, that's different. All right, you'll have a week. If you need longer than that, you'll have more than that. You can see, I'm not that worried about deadlines. As long as it's done by the end of the semester, I don't honestly care that much. I just have to be able to submit grades on time. So medulla oblongata. So I will give you at least a week and 2 weeks if you want it. It doesn't matter to me. The medulla, I'll give you 2 weeks. I'll give you 2 weeks. The medulla oblongata contains nuclei that house the respiratory and cardiovascular centers. So in addition to the corticospinal tract, It's going to house the center that's going to control the rate and force of the heartbeat, the diameter of the vessels, so whether vessels vasoconstrict or vasodilate. The medullary rhythmicity center, which is going to control the rate and rhythm of breathing. The vomiting, coughing, hiccuping, deglutition, which is swallowing, and sneezing centers are also located here. And a bunch of other nuclei that we won't discuss. So this is controlling a lot of your basic life functions, your heartbeat, your breathing. If you think about it, this is why babies with anencephaly, meaning that they're born without technically having a brain, can survive until after birth and survive for a little while afterwards, most often, is because their brain stem is intact. This is also, as I told you before, this is where opioids act on. If you heard, and I've never been a fan of the Foo Fighters, but their drummer just recently passed away. He was found dead in a hotel room. Looking at what I saw, and I can tell you, I honestly have never really listened to the Foo Fighters. But I think anyone with a musical talent should be appreciated. And if he's gotten to that point, he has to be talented. Opioids are acting on these centers. So opioids are targeting the medulla oblongata. I told you really briefly that when I broke my jaw, I had a morphine overdose. And I remember some of it, and then I remember just losing consciousness and coming to and being very disoriented and couldn't remember where I was and how I'd gotten there or who was in the room with me. And what it does is it essentially slows down your breathing and the extent of your breathing until you stop breathing altogether.
That drummer, when you brought that up last week, I wanted to mention it, but we were busy. You said you assumed that he overdosed.
Did he not?
He sure did. Oh yeah, no, he did.
Okay, because I don't want to
split something funny, but
correct.
They found over 10 different substances
I
in
heard
his
that.
system. Yeah, opioids, benzodiazepines, antidepressants, marijuana, a lot.
Well, and I don't really, I'm one of those people where I try really hard not to judge what anyone else is dealing with.
Yeah.
Because, to each his own, and looking at my caffeine addiction, I really can't talk. But, and I don't know about the stress level that they deal with. I look at things like, who's a Prince Harry, who's now living in California with his wife. You'll remember, of Great Britain. And I just think, media, leave him alone. He's trying to do the right thing by his wife and his marriage. Leave the guy alone. But you notice that he showed up at a ball game without his wife, and all of the paparazzi decided they were having marital problems. People, they have 2 little kids at home. Just because she maybe wanted to stay home doesn't mean they're having marital problems. So yeah, I don't really understand that, but it's sad. But that's why it's so scary, because we're realizing what opioid addictions are and how nasty they are. And they're acting a lot on neurotransmitters too. And that's the reason they're so stinking addictive. Yeah, I'm glad I wasn't wrong because I was just reading stuff on Facebook about it. That's the only reason I knew about it. The pons is a bridge that connects the spinal cord with the brain. The anterior bulge in the brainstem that's rostral to the medulla oblongata, and it's going to develop from the metencephalon. It consists of nuclei. Remember what nuclei are when we're talking about the central nervous system. They're clusters of cell bodies. It relays nerve impulses related to voluntary skeletal movements and coordination from the cerebral cortex to the cerebellum, and also contains the pneumotaxic center that controls breathing rate. This, in addition to the medulla oblongata, is another place where opioids act, is on the pneumotaxic center because it's going to slow down breathing. It contains the cranial nerves 5, 6, 7, and 8. So on they traveled, trigeminal and abducens, found facial, Voldemort, vestibular cochlear. You guys, if you haven't figured out, I really want you to memorize the cranial nerves. It's important. And it'll make you look smart later on too.
Can you repeat that? Because what I had was on they traveled by the morgue, and then you said on they traveled and
all right, let me write it down. Let me type it out. Because that on the morgue, no, they didn't travel by the morgue. Let's see here. That was funny. So we'll talk about this again, but if I write this down right here, I don't have to write it down later. They traveled as I can't type, traveled and this is the mnemonic I use because I like Harry Potter and I can remember it for that reason. And they, so on they traveled and found Voldemort, or he who should not be named, Voldemort, guarding the very ancient Horcruxes. If you remember, if you read the 7th book of Harry Potter, this is a major part of the book. Is it says on they traveled and found Voldemort guarding very ancient Horcruxes. So olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory or spinal accessory, hypoglossal. if you like other things, and we'll come back to it. I'll put it up again. But if you like other things, such as fashion, which isn't really I like fashion, but I don't like mall shopping. So this doesn't really work for me. But if you come in and you look up cranial mnemonics, for cranial nerves, 40 tips on mnemonics and remembering the cranial nerves. All right, we're not going in through that one because I don't want to give my computer a virus. Give me 1 second. There's more. Only one of the 2 athletes felt very good, victorious and healthy. Some say money matters. Some say marry money, but my brother says big brains matter more. Only one of the, oh, okay, we just read that one. I know that there's one about Abercrombie and Fitch. And it might be under that site that this computer doesn't have all the viral protective stuff I would like for it to have. And so I'm not going to open that up. This is one that my old research advisor really knew, was on old Olympus's towering top, a Finn and German viewed some hops. That doesn't work so well for me because I'm not that, I just don't have, I don't get it. I do, but I don't. So lots of different mnemonics. If I was at a school, because they have incredible software, I would open that one site because I'm guessing that's where the Abercrombie and Fitch. Well, actually, let's try this. And the only reason I mentioned this Abercrombie and Fitch was because I remember when I was first teaching AMP, one of the students had this written on the top of their test. And you can see that I don't remember it completely. That's why I have to look it up. Okay, so here's a bunch of them. Yeah. So there's 35 different ones for you. And with the Harry Potter one at the very top. So find one that works for you. So the midbrain is going to extend from the pons to the diencephalon. The mesencephalon becomes one mature structure of the midbrain. So it's really the, well, okay, the telencephalon just becomes the two cerebral hemispheres. But the mesencephalon, rather than becoming multiple structures, is going to become one. It's going to become the midbrain. I like to think of mesen as being the middle. So if I think of mesen as being a middle structure, then I remember this is a midbrain, because midbrain meaning middle of the ring. Contains a cerebral aqueduct and is surrounded by gray matter that's involved in controlling pain. It contains the motor nuclei of two cranial nerves that control eye movements, the olfactory nerve, cranial nerve number 3, and cranial nerve number 4, the trochlear nerve. The cerebral peduncles, or little feet, are structures at the front of the brain that arise, these little structures right here, that arise from the front of the pons and contain large ascending sensory and motor tracts that run to and from the cerebrum and the pons. So they look like 2 feet holding up the cerebrum. That's where the name comes from. The midbrain also contains the corpora quadrumana. Remember, this is 4 bumps, not 3 bumps, 4 bumps. The superior colliculi and the inferior colliculi. These are 2 structures that I was actually able to find on our little tiny sheep brain, because they're pretty obvious. 4 bumps in 2 rows. The superior colliculi is going to serve as a reflex center for movements of the head, eyes, and trunk in response to visual stimuli, allowing you to be able to watch something as you move away from it. Or when you're in your car and you're coming up on a stop sign or a street light, being able to focus on the street light as you move towards it. That's a visual reflex. Inferior colliculi, as I scared my poor dog on my lap, is centers that startle you in response to loud noises. So they're going to serve as reflexes for movements of head, eyes, and trunk in response to an auditory stimuli, such as a startle reflex. When you hear something really loud and you move your head in that direction, that's going to be controlled by the inferior colliculi. I think of it as your eyes are a little bit above your ears. So the superior colliculi are going to control the visual stimuli because they're up on top. And the inferior colliculi is going to control the auditory stimuli because your ears are a little lower down than your eyes. The midbrain also contains a substantia nigra, which produces dopamine. It's responsible for relaying messages that plan and control body movement. It also is going to relay, and this is crucial, it's going to relay inhibitory signals to the thalamus and basal ganglia to suppress any unwanted body movement, like tremors. any unwanted unconscious movement is going to be suppressed. Parkinson's disease is a degenerative progressive disorder that affects the neurons of the substantia nigra and basal ganglia, in that it's going to reduce the levels of dopamine that are secreted by the substantia nigra. This disorder results in shaking, stiffness, and difficulty walking, balance, and coordination, because you can no longer suppress unwanted body movements. Rather than having your muscles and your limbs be flexible like they always are, unless you want them to be stiff, you no longer are able to control that as well. Which statement is false about the medulla oblongata?
Is it B?
B as in Bravo?
Yes.
Yes. Because first off, we know that white matter is not containing synapses. So yes. Good job, Sam. Sam, I can't remember if you like to be called Samantha or Sam.
Sam.
Sam, okay, I thought about that when I was responding to your email earlier. I thought I can't remember. Reticular formation, a loose web of gray matter. Remember what gray matter is. Gray matter is synapses, cell bodies, and dendrites. A loose web of gray matter that runs vertically through all levels of the brainstem. It's going to regulate muscle tension, maintain tone, balance, and posture. Neurons within the reticular formation have ascending and descending functions. The ascending function is the reticular activating system, or RAS. It's going to alert the cortex to incoming sensory signals. It's responsible for helping you maintain consciousness and awakening from sleep. This is why when your room starts to get lighter, you start to wake up, generally. I read something recently, or saw something recently, that said that sleeping with any light in your room is actually not good for you. And I don't know if you're like me, where you like to sleep in solid dark, which I do, because I sleep better. I would guess that it's related to this.
I wonder if it's related, like, I work.
Say it again.
So I have blackout curtains and a mask, but it's still never possible for me to sleep when it's completely dark. Unless
When it's not completely dark?
Yeah, I mean, even with a mask and blackout curtains, I feel like, it never gets as dark as it would at nighttime with the same stuff.
Huh.
I know that your brain doesn't produce as much melatonin during the day.
Right.
So, but I wonder if that's similar too. Like, even if you try to falsely, create that with curtains and masks and stuff, it's never quite the same, is it, as sleeping at night when it's dark?
I think it's because your reticular formation, and that it's also it acts on the pineal gland, which is secreting melatonin. So that's the reason during daylight savings, we all get messed up and get tired when it's earlier because it's darker outside. But yeah, there was something, I'd have to go back and look at it again, but it made the comment that it's actually, it's bad for you. And I know it can cause stress. But I think part of that is watching TV when you fall asleep is bad for anxiety levels. Inactivation of the reticular activation system, or RAS, produces sleep. Well, damage to the RAS can result in a coma. That irreversible sleep. The cerebellum, and you guys, I'm going to pause for 1 second. We're going to watch a video and then come back and take a 5-minute break. But I want to stop just for a minute because I can tell you that of all the things that I worked to explain, the cerebellum is probably one of the harder things for me to explain. I know that it has to do with constant readjustment for movements, but I found a link. Oh shoot. Oh no, you know what? I know where it is. I thought now I'm looking at a different course and I didn't attach it to this course. But let's open a different link. So this is functions of the cerebellum. And so
friends would walk up to me and just be like, what the f is in your butt?
So one thing that it's constantly reassessing your movement.
Hi everybody, Dr. Mike.
Constantly reassessing your movement and adjusting your muscles and the tension of your muscles. As you go to start an activity, it's assessing whether that object is heavy, light, and adjusting how much muscle contraction you put into it. If you have to activate other muscles when you realize that it's heavier than you thought it was, the cerebellum is what's going to be involved in reassessing things.
Okay, in this video, we're going to take a look at the cerebellum. Now, to begin with, I want you to think about this scenario. You're going to a friend's place to help them move. They point to the corner of the house where there's a box. And they say, hey, can you pick that box up, chuck it in the truck for me? Now, they say to you, look, this box is heavy, so be careful. Now, as you're walking over to the box, you're thinking, okay, it's a heavy box. I'm going to have to utilize all my muscles, bend with my knees, not with my back. You get over there, you grab the box, and then you pick the box up, and you realize that the box is actually empty. Now, what is stopping you from flipping yourself backwards? Because you've prepared yourself to pick up a really heavy box. Well,
so your body's already reassessing what's going to be involved. And then the reality is different than what you thought it was going to be.
The thing that stops you from flipping back is your cerebellum. What the cerebellum does is it takes your intended movement and then it takes the actual movement and then it calculates the error between those two. Once it calculates that error, it then fine-tunes that mode of movement. So what the cerebellum does is it fine-tunes mode of movement, and it does it through a couple of different functions. So while it does fine-tune mode of movement, you could divide what the cerebellum does into, it helps fine-tune muscle tone.
So tone would be contraction. How much contraction is necessary? How much force is necessary? And coordinating those different muscles so that it's a smooth contraction.
Coordination and balance and posture. These are the main functions of the cerebellum: muscle tone, coordination, balance, and posture. Now, before we go into more detail about the function, let's talk about the anatomy a little bit. So the cerebellum, we've drawn up here, we've got the cerebrum, which is the main part of our brain, two cerebral hemispheres. This is a lateral view. So we've got the cerebrum, we've got the brain stem, which is midbrain, pons, medulla, and we've got the cerebellum sitting here. So you can see the cerebellum sits inferior to the cerebrum, posterior to the brain stem, and the part of the skull that it sits within is the posterior cranial fossa, and it's separated from the occipital lobe of the cerebrum by the tentorium cerebelli. So if we were to take the cerebellum and the brainstem and pull it out and have a look at it, we've performed a sagittal view, so we're looking into it from the side, from a lateral view, and we can see a couple of things. Firstly, it is folded like the brain, like the cerebrum itself. So it does have these gyri and sulci, bump up, dips down, and so forth. It does have a cortex, an outer region, and a medulla, again, like the cerebrum. But a couple of things is that if you were to look at it under a section, it looks like there is a branching, like a tree that comes out from this aspect.
What's that tree called? That branching?
Arbor vitae.
Good job. Yes, Ivy. Perfect.
The brainstem, and that's called the arbor vitae, which means tree of life. And you can also see we've got the brainstem here with the midbrain, pons, and the medulla, and you can see part of one of the ventricles here. So if we were to divide the cerebellum up anatomically, you can divide it into three lobes. So these three lobes, first of which, we've got the anterior lobe. Second is all this here is the posterior lobe. And then this lobe underneath here, this is called the flocculonodular lobe. Great name, isn't it? Flocculonodular lobe. These are the three anatomical lobes. You can see the flocculonodular lobe sits underneath. So if we were to take the cerebellum, and look at it from a posterior view, like we've done here, what we've actually done is we've taken that flocculonodular lobe and we've pulled it out so we can see it. So this is the flocculonodular lobe here. The flocculonodular lobe sitting under here. Now, if we were to find the anterior and posterior lobes, we could very roughly separate them out like this, where we've got the anterior and posterior lobes. Now, again, these are anatomical divisions, right? 1, 2, 3. But if we were to look at it functionally, we can actually separate the cerebellum a little bit differently. Firstly, we're going to begin with the flocculonodular lobe. And the reason why is because the flocculonodular lobe anatomically is distinct, but also functionally it's quite distinct. And the reason why is because it deals predominantly with vestibular movement. So this is referring to the vestibular system. So balance. So I want you to think about the vestibular system.
You guys, we haven't talked about that, but when we are going to talk about a little bit about it tomorrow, Wednesday night. But the vestibulocochlear nerve, cochlear meaning hearing, vestibulo meaning balance. So when he's talking about the vestibular system, he's talking about balance, which happens in your inner ear via the cochlear nerve.
Information coming from the vestibular cochlear nerve, right, from the inner ear. This is cranial nerve 8. So information coming from your balance areas of your inner ear is sending information to the flocculonodular lobe. So I could draw up an arrow here and say that information from cranial nerve 8, which is the vestibular cochlear nerve, is going to the flocculonodular lobe. And so another name for this area is the vestibulocerebellum. So this can also be recognized as the vestibulocerebellum.
I like him because I learned about things that I didn't know. Like I can tell you that I don't think I've ever talked about the flocculonodulum. But it makes sense if I think about it.
Vestibulocerebellum. And what does it deal with? It predominantly, if it's going to be cranial nerve 8, it's going to be dealing with balance and posture. What did I say? Balance and posture. So let's write this down. I'll just write balance. Okay. Now, when we move up, what you're going to find is that when we to break these anatomical lobes, the anterior and posterior, up into their functional areas, we don't do it in this way. We actually do it in this more vertical way. And this is how it works. Firstly, you can see that there's 2 hemispheres of the cerebellum, and it's separated out or connected by this area in the middle called the vermis. So let's actually label that. The vermis.
So the vermis would be like the corpus callosum, connecting the 2 hemispheres of the cerebrum. So remember, they call the cerebellum the little brain, cerebrum the big brain. So the corpus callosum and the vermis are analogous in their function.
Now, importantly, the vermis and its areas immediately next door which are called the intermediate zones. Intermediate zones. The vermis and the intermediate zones are its own functional area, right? Now, let me tell you why. Because there is actually a topographical map of the body placed on the vermis and the intermediate zones. Just like we've got the somatosensory homunculus and motor homunculus on our cerebellum, we have a homunculus on the vermis and intermediate zones. So let's have a look. What we've got is it looks a little bit like this.
He's going into a lot more detail than I expected. But the homunculus, we'll see that again in a second. Homunculus.
A little bit like an alien. It's upside down, right? And here are the eyes. Now we've got one here as well in this position. So what can you see? First thing is the axial musculature. So the musculature of the neck and the trunk, the head, neck, trunk, for example, all lies within the vermis. So that means when it comes to fine-tuning, coordinating muscle tone of the head, neck, and trunk, it's all coming from the vermis. When we deal with coordinating and fine-tuning the musculature of the limbs, this is all coming from the intermediate zone, right? That's important. Okay, another thing is that information, we said information coming from the vestibulocochlear nerve is going to here to do with balance. Information coming from these parts of the body is going to be proprioception, right? So telling you where your arms are in their space, telling you how bent a joint is, telling you how contracted a muscle is. All this type of proprioceptive information from the limbs, intermediate zone, and from the trunk, head, neck, and trunk, vermis. It's coming from the proprioceptors to these areas, right? So I can draw up an arrow and say proprioception. So proprioception is going to these areas here. For the limbs there, and for the head, neck, and trunk there. All right. Now, well, the other thing is that this flocculonodular zone is also known as the vestibulocerebellum. This area has another name. It's called the spinocerebellum because it's all coming through the spinal nerves to go to these areas. Right? So let's rename it the spinocerebellum. The spinocerebellum is also this area here, including the vermis and intermediate zones. All right, the last area are these most lateralized areas. And these most lateralized areas, I'll first give you the name, is called cerebrocerebellum. And that should tell you where it's getting its information from. It's getting it from the cerebrum, from the cerebral cortices. So this area deals with very fine tuning of motor information, skillful tasks, playing the piano, doing very important tasks like that. Right there, very skillful tasks. So what we find is that information coming in to.
So when you're like Justin, we know how smart Ivy is, and it probably took her absolutely no time at all to get perfect handwriting. But when you work on training your hands to write your letters, you're essentially training your cerebellum. When you're working to learn, to color within the lines, which hopefully we don't always color within the lines and everything. But when you're learning to color within the lines, you're training your cerebellum. You only color so far. You don't color beyond this line. Oh, perfect. Etch-a-sketch.
These most lateralized regions are coming from the primary motor cortex.
Where have you seen that before? Remember, that's found in the same cortex is found on right anterior to the central sulcus in the brain. It's got the same name.
The premotor cortex.
Remember that that's right anterior to the primary motor cortex in the frontal lobe.
Now, together, you got planning and actually thinking about doing that motor movement. That's coming in here, so it can be fine-tuned, right? It's going to have information coming in from the primary and secondary somatosensory cortex, so information about.
And that cortex is on the post-central gyrus, so right posterior to the central sulcus. So we have these same areas in the cerebellum.
Sensation. This is important because we need to know information about the musculature in the body to make a decision, and information coming from the visual system, specifically about objects that are moving in our vision, is all coming into this cerebrocerebellum, these lateralized areas. All right, really important. So overall, what we're going to find is this. I said balance and posture, vestibulocerebellum. Coordination is going to be mainly here because it's really skillful tasks, right? Coming from the motor, premotor, somatosensory, visual, and tone, how contracted or relaxed the muscle is going to be coming from the vermis and intermediate zones, and so we can pop.
Whether you pick up a pencil to write with it or you pick up a pencil, and break it, is going to be the cerebellum. How hard do you grip that pencil?
Tone in this area here, and this is how we can functionally divide the cerebellum. So hopefully this gives you a nice summary of the anatomy of the cerebellum and the physiology.
Okay, that was a lot, and I can tell you that I learned things with that one that I didn't plan on to. So let's take 5 minutes and come back at 7:55, as my clock says right now, and we'll continue forward. I'm gonna charge my phone because I'm gonna listen to it. I can utilize my time by listening to an explanation about membrane potentials for my class tomorrow. Like I did listen. Oh. Houses here. How's that? How's that reach? I think that's the case with most of what goes on with them because they have high pain tolerances. No, you're staying. Stay. Yeah, you're a good boy. You're a good boy. You're a good boy.
sit there in the middle of the floor.
You said you went outside without me. I need a monitor.
Yeah, there you go. Thank
You
you for
left me
getting
with-
up and letting me walk home.
You left me with her. What were you thinking? Okay. So let's jump into this again. And we'll make as much headway as we can. And then we'll probably work on part of it on Wednesday night also. So the cerebellum, largest part of the brain as a whole, second largest part, I'm sorry, contains more than half of all the brain neurons. So about 100 billion. Consists of right and left cerebral hemispheres connected by a vermis. The superficial cortex of the gray matter with folds, the folia, branching with the white matter, the arbor vitae that Ivy knew about. Yep. The deep nuclei. The deep nuclei. So remember, deep nuclei is going to be gray matter, because nuclei are cell bodies, and in the central nervous system, and also in other places, same as the peripheral nervous system, cell bodies are always going to be gray matter, because gray matter meaning non-myelinated. So deep nuclei that make up the gray matter and give rise to nerve fibers that carry impulses from the cerebellum to other parts of the brain and spinal cord. It also has several sensory, linguistic, emotional, and other non-motor functions, including comparing the textures of objects, perceiving space, as tested with pegboard puzzles, recognizing objects from different views, keeping judge of elapsed time and maintaining a tapping rhythm, helping direct eye movements that compensate for head movements. So allowing you to keep your gaze fixed on keep your gaze keeping your gaze fixed on an object. Judging the pitch of tones and distinguishing between similar spoken words. Helping in verbal association tasks. Planning and scheduling emotional control. Many hyperactive kids have smaller cerebellums. So very, very involved. We're going to come back to it here in a moment, but we're going to talk about some other things first. The hippocampus is critical for forming new memories and spatial orientation. If you look at a lot of this, you'll see that a lot of these functions are interrelated. There's other areas of the brain that have the same function. Remember when we were talking the other night about neural integration? That's what we're looking at here. So the hippocampus learns from sensory input while an experience is still happening, but it has a very short-term memory. However, later, perhaps during sleep, your hippocampus will play that memory repeatedly over and over again to your cerebral cortex, which is a slow learner, but forms longer lasting memories. This is why studying right before you go to bed is great. is because so much of what you studied right before you go to bed is going to be replayed back to your cerebral cortex. An example of a relationship between the hippocampus and learning. A man underwent surgical removal of a large portion of both the temporal lobes and both hippocampi in order to treat severe epilepsy. He was left unable to establish the memories. He worked with the same psychologist for more than 40 years after his operation, but could not remember who she was from day to day. Can imagine what that would be like. I don't think I can imagine it. Alzheimer's disease. The hippocampus tends to show the most rapid loss of tissue early in the disease. The shrinkage is responsible for the short-term memory loss that occurs with Alzheimer's. Their cerebral cortex is not altered. Long-term memories are going to stay intact. Or many of them will stay intact. Memories about what happened when you were 10 will be remembered. Things that happened 4 hours ago might not. The diencephalon is comprised of 3 regions. The epithalamus, which includes the pineal gland, which secretes melatonin. The hypothalamus, the thalamus and the hypothalamus. Remember, the thalamus is that grand relay station. The hypothalamus is the major coordinator of the autonomic nervous system. So the thalamus is that gateway. Each cerebral hemisphere contains half of the thalamus and is connected by an intermediate mass. It's the grand relay station. Remember when we were talking about sensory fibers. Sensory fibers are going to synapse in the thalamic nuclei and be relayed to a particular area of the cerebral cortex to be interpreted. Motor information, remember the higher and the lower spinal nerves, are going to be relayed through the thalamus to the primary motor cortex, right here, primary motor cortex. Primary motor area of the cerebral cortex. The thalamus, in addition to being this relay station, is involved in memory and emotional functions of the limbic system. I don't want you to learn all these different groups and areas and what they specifically do. We may learn later that we don't really know this as well as we think, and that it's not mapped out as easily as they make it look here. That's why I don't care if you memorize this. So it's a complex of structures, including some cerebral cortex of the temporal lobe and frontal lobes, and some of the anterior thalamic nuclei. The epithalamus contains a pineal gland which secretes melatonin. And melatonin is a hormone that's going to regulate day and night cycles. More melatonin is released during the darkness than the light, which promotes sleepiness. And like Christina said, and what we were trying, I think we were both getting at, is when things become darker, your reticular activating system is going to be involved in activating your pineal gland to secrete melatonin. And what I would be curious about is I know that one of the major side effects to taking melatonin supplements is migraines. I quit taking it for that reason.
It can also give you really vivid dreams too.
Say it again.
It can give you really vivid dreams.
Really weird dreams, vivid dreams.
Or vivid, yeah.
Okay, another reason I'm glad I don't take it anymore. I don't need to have weirder dreams than I sometimes have. I would be really curious why it causes migraines, and maybe I'll have to look into it at some point. So hypothalamus controls hormone secretion. So this is the major control center of the endocrine system, meaning your hormones, and regulating the autonomic nervous system. So it controls hormone secretion by regulating secretions from the anterior pituitary. And the anterior pituitary secretes hormones involved in regulating growth, metabolism, reproduction, and stress responses. The posterior pituitary regulating labor contractions, lactation, and water conservation. Sam's guy with the osmolarity issues. One of the major issues with not being able to concentrate, maintain your osmolarity, it has to do with water conservation coming from your posterior pituitary, which when you talk about renal function in 122, you'll learn all about. But the hypothalamus is attached to the pituitary via a stalk called the infundibulum. Oh, great name, infundibulum. So it's a major control center of the autonomic nervous system in that it's going to influence heart rate, blood pressure, gastrointestinal secretions, motility, hunger, thirst, body temperature, and etc. If you remember, a lot of these are also controlled by the medulla oblongata. So they're working together. Heart rate, blood pressure. That's also the medulla oblongata. The mammillary bodies got their name because they look like little tiny boobs right here. Two small rounded projections that serve as relay stations for reflexes related to smell and taste. You guys remember, these are also part of the limbic system. I mentioned the experience, and I think Elizabeth mentioned the experience about smells from her grandma's house. And I told you how I don't like the taste of coffee, but I love the smell of it because of memories that it brings. So the mammillary bodies are also involved in your memories with regards to smell and taste. Which of the following regions is a major convergence area for most sensory input before it's sent onto the cerebral cortex? What do you think? Someone? I wonder if Sam's trying to talk, but she's muted.
Oh no, I was trying to figure it out, and I was kind of talking to my kid trying to figure it out. I was thinking maybe is it A?
It's not. A is more the autonomic. What else? What's the grand relay station?
The hippocampus.
Nope. That's
No?
learning. It's short-term memories.
Zero. Thalamus.
It's a thalamus. Yep, it's a thalamus. C. So surface anatomy of the brain. Cerebral hemispheres, the cerebellum, the brain stem, the gyri, meaning the folds, the top folds, the sulci or fissures. Sulci are smaller fissures. That's the way I think of it. Longitudinal fissure, the central sulcus, anterior, posterior. So pre-central gyrus, post-central gyrus, pre-central gyrus, post-central gyrus, the lateral sulcus.
Professor Gessel, that's where I struggle, like looking at the diagram.
Can you say it a little bit louder, Christina?
Sorry, I struggle with knowing if the questions are asking for, like a gyrus or succulus, or if it's just asking for a lobe, because some of the questions have both mixed together, and I feel like it can be kind of hard to tell with just the way the arrows are touching.
If it asks, let me, I can't remember exactly how the questions are asked. But if it asks about functional anatomy, it's asking for areas. Like Broca's area, Wernicke's area, the primary motor cortex. If it's asking for structural anatomy, it's asking for the precentral gyrus, the postcentral gyrus, the frontal lobe, the occipital lobe, the temporal lobe. Does that help?
A little bit. I think I just have to keep looking and, seeing different versions of the same diagrams. It's definitely harder when it's all one color, but I realize in real life, they're not different colors.
Yeah, I understand. Darn it. I will do my best to help as we go. So central sulcus right here, the precentral gyrus, the postcentral gyrus. And one thing that you're probably struggling with more with this one, that you're not struggling with other ones, like the one that we did on part of the spinal cord, and you won't struggle with the special senses, is that I've taken the diagrams from the manual and adjusted them and put in the right answers. And I can tell you it takes hours for me to do that. But now that I know how to do it, and it's harder for me to do it for this class because I already had those PowerPoints set up. And I wish I had 8 hours more in a week that I could spend readjusting those figures. But you won't have that problem with the special senses work, because all of the diagrams I've adjusted and giving you the answers. So Christina, sir, what I'm saying so much in a shorter way, more concise way, survive this one and recognize that I'm going to give you the answers for the special senses.
All right, thank you.
You're welcome. So the precentral gyrus, the central sulcus, the postcentral gyrus, the lateral sulcus, the pons, and then just inferior to the pons, the medulla oblongata, and the spinal cord, and the cerebellum. The cerebrum, the largest and most conspicuous part of the human brain, the site of sensory perception, memory, thought, judgment, and voluntary motor actions. Each hemisphere has 5 lobes. The frontal lobe, which includes the precentral gyrus, the parietal lobe, which includes the postcentral gyrus, and they're separated by the central sulcus. The occipital lobe, the temporal lobe that's separated by the lateral sulcus, and the insula, which is deep, which I'm not sure if anyone has ever dissected out the insula. I don't know if we really even know the anatomic boundaries of it. It's the least understood. Summary of the different functions. Frontal lobe, abstract thought, explicit memory, mood, motivation, foresight and planning, decision-making, emotional control, social judgment, voluntary motor control, speech production, that voluntary motor control coming from the precentral gyrus, the primary motor cortex. And then the motor association area and the premotor cortex. The insula, taste, pain, visceral sensation, so sensation around the organs, internal organs, consciousness. Maybe the part of this may be really close, because talking about consciousness, the pineal gland. Remember how it's kind of deep? Emotion and empathy, cardiovascular homeostasis. Those internal parts like the medulla oblongata. Parietal lobe, taste, somatic sensation. So skin, muscles, joints, bones. Sensory integration, visual processing, spatial perception, language processing, Wernicke's areas in here. Numerical awareness, visual awareness and visual processing, occipital lobe working with the temporal lobe on visual processing. The temporal lobe, hearing, smell, emotion, learning, language comprehension, another Wernicke's area again. Memory, consolidation, verbal memory, visual and auditory memory, and language. The 2 hemispheres are connected by a white fibrous tract, the corpus callosum. I told you about my friend and his kids that were all born with corpus callosum agenesis, meaning they didn't have a corpus callosum formed. And none of them, the oldest one living child that he had was 16. And all of his other children had died. And the child that he had was almost a nonverbal cerebral palsy, sort of symptoms similar to that. So you can understand why that connection is so important. Cerebral white matter. Remember what tracks are. Tracks are bundles of axons in the central nervous system. So most of the volume of the cerebrum is white matter, glia and myelinated nerve fibers that transmit signals. The tracks are bundles of nerve fibers in the central nervous system. Projection tracks are tracks that are extending vertically from lower brain and spinal cord centers into the cerebral cortex. So a major projection track is the corticospinal tracts, those voluntary motor tracts of the spinal cord extend up to the cerebral cortex via these projection tracts. Commissural tracts. A good way of thinking of commissural tracts, they're what are going to make up the corpus callosum. So axons that cross from one hemisphere to the other. Association tracts, rather than being projection, meaning they're coming from the spinal cord and the brain, or commissural coming set between the two hemispheres. Association are connecting different regions within the same hemisphere. So long fibers are going to connect different lobes within the hemisphere. Short fibers are going to connect different gyri within the same lobe. Remember what gyri are, the top folds. Any questions so far? Are we doing all right? Cerebral cortex or neocortex, gray matter covers the surfaces of the hemispheres. Neural integration takes place in the gray matter of the cerebrum. Remember, this is where we talked about neural integration before. The more synapses and the more dendrites a neuron has, the more abilities it has to integrate information, take in and process the information, and decide what to do with it. So the gray matter is where this is occurring in the cortex, cerebrum. So the cerebral gray matter is composed of neuronal cell bodies and dendrites, containing 14 to 16 billion neurons. During embryonic development, that gray matter of the brain is going to develop faster than the white matter, resulting in a cortical region that rolls and folds on itself. You guys, please stop me if you have questions. The cerebral cortex contains two types of neurons. Stellate cells, stellate meaning star-like shaped, and pyramidal cells, meaning they're more triangular in their cell bodies shape. Stellate cells have spheroidal somas with dendrites projecting in all directions. They look like little stars. Or with me, I'm going to zoom in just a little bit. They look like little stars. They receive sensory input and process that information on a local level. The destination of these impulses is going to help to localize somatic sensation. Pyramidal cells are going to extend from the cerebral white matter and conduct impulses for motor commands. So stellate cells are sensory, pyramidal cells are motor. And they are going to carry impulses that result in the contraction of the biceps brachii, for example. You guys, if it's conducting impulses to the motor commands, where do you think you see a lot of these also? Watch that big long video. What was he mainly talking about? What was a major part of what he was talking about?
Cerebellum.
Yeah. You're going to see a lot of pyramidal cells in the cerebellum. Exactly. Good job, Eliza. The basal nuclei or ganglia. So this is, we kind of talked about this before, but I don't think the repetition is bad. We're just we're relating it to other things. So this is what's affected by Parkinson's disease with the substance. Louie, buddy, oi. You never know when we had a wind monster outside. Louie, it's all right, buddy. Yeah, you're a good boy, protecting us. I would hide from her too. He's here hiding underneath my table from my mother. I would hide from her too. I understand. Okay, so basal nuclei or ganglia, masses of cerebral gray matter buried deep in the white matter that are involved in motor control. Receiving input from the substantia nigra in the midbrain and motor areas of the cortex and relaying those signals back to the rest of the body. Because of the integration, I don't think it's bad that this stuff is scattered throughout the PowerPoint, talking about certain places. I can't remember why I put it together the way I did, but I kind of like it because we're looking at a lot of integration. Which of the following is a function of the basal nuclei?
Charlie, help initiate and terminate.
I agree with you. So if you remember, one thing that happens with Parkinson's is not being able to terminate movements. Not having a movement go over well beyond what you want it to do. So cerebellum and motor coordination aids in learning motor skills and maintains muscle tone and posture and smooths muscle contractions. If you want to know a lot about the cerebellum more than we talk about here, go back and watch that video again, because he did a really great job with that one. Coordinates eye and body movements, coordinates the motion of different joints, and functions as a comparator in motor control. How much motor control do you need to put into an effort? The cerebellum is going to receive information from the motor cortex, brainstem, and spinal cord. And it's also going to provide information back to the motor cortex, brainstem, and spinal cord. Controlling muscles by using sensory feedback from receptors. The cerebellum in Purkinje cells, one thing that we haven't talked about, Purkinje cells found in the cerebellum are going to assist in motor coordination by correcting muscle performance with the purpose of the movement, making it smooth. Working with the reticular formation, your eyes, your inner ear meaning posture and balance. And the premotor cortex and primary motor cortex and motor association cortex. Taking that information in and smoothing the muscle performance. Also sending information to the primary motor cortex areas and premotor cortex and motor association cortexes. And sending that information down to the spinal cord and to your joints and your muscles to maintain contraction and at the same time maintain your posture so you don't fall over. Lesions in the cerebellum can cause ataxia, and some tasks such as walking up the stairs can become impossible. Can cause difficulty walking or poor balance, tremors, slurred speech when you can't coordinate your mouth and your tongue muscles, eye movement abnormalities, lack of coordination, trouble eating and swallowing, heart problems, deterioration of fine motor skills. Remember I said that one way when we're training our fine motor skills, we're training our cerebellum. Our gait abnormalities, chronic alcohol abuse can cause cerebellar degeneration or atrophy, typically existing as an ataxia, meaning a lack of coordination of lower joints and trunk instability. Normal cerebral cortex, normal cerebellum, atrophy of the cerebellum.
Is that what's referred to as wet brain?
Wet brain? I
Okay.
don't know. Mother? Yep. What's wet brain? Wet brain. She doesn't know either.
Well, from it's just a layman's term, meaning that the alcoholic has done so much damage to the brain that they now, they appear to be drunk even when they haven't consumed alcohol. So it
I'll
would
bet
be
you
this-
money that's it. Yes. I'll look into it, but yes. The limbic system, so stay with me for 5 more minutes and we'll just see how far we get along. The limbic system is an important center for emotion and learning. Found in each of the hemispheres, the prominent components are the cingulate gyrus, which arches over the corpus callosum, and the frontal and parietal lobes. The hippocampus, remember this is short-term memories, and the amygdala, which is immediately rostral to the hippocampus, which is involved in emotion. This is why teachers like to use emotion when they teach, is because students tend to remember if you can affect their emotions. Me mentioning that moment when that professor shared that mnemonic about the cranial nerves that I'm not going to share because my mother's here. You won't forget it once you hear it. The limbic system, an important center for emotional learning, contains centers for gratification, senses of pleasure or reward or aversion, senses of sorrow or fear. The amygdala will receive information from general and special senses and mediate emotional responses to such stimuli, such as a disgusting odor, a foul taste, a beautiful sight, or pleasant music. I told you about my PTSD of smelling those insulator cozies, because your limbic system is so connected to your emotions. And you learn to associate something with a specific smell, taste, or sight. Integrated functions of the brain, higher brain functions. Sleep, memory, cognition, motor control and language, involving interactions between the cerebral cortex, the basal nuclei, the brainstem, and the cerebellum. Like we saw when we were looking at the lobes, the functions of the brain don't have easily defined anatomical boundaries. I told you I don't know if they've ever dissected out where the insula lobe is specifically and how it's separate from the other lobes. Integrated functions of the brain focus mainly on the cerebrum, but involve combined actions of multiple brain levels. Electroencephalogram. Monitoring surface electrical activity of brain waves. Millions of brain neurons close to the surface of the cerebral cortex are collectively creating action potentials that are detected by electrodes positioned on the scalp. They're useful for studying normal brain functions such as sleep and consciousness, and from that being able to diagnose degenerative brain diseases, metabolic abnormalities, brain tumors, etc. A lack of brain waves is a common criterion of brain death. So, you know what? Let's just end right here. So, we will discuss the special senses. I'm going to spend more time on anatomy and less time on function, because I know you have videos and animations that you can watch to find the answers for the functions. But we will spend some time talking about them, specifically hearing, balance, and sight. And then I'd like to go forward with this chapter, because it's all going to coordinate together. And I hope you have a wonderful night, okay?
Thank you, you too.
Thank you. Good night.
Night.
You as well.
Thank you.
Bye.
Bye.
Professor Gissel, I have a quick question. If my son was wondering if your brain fell into your foramen magnum, could you survive?
Probably not. I told you about my good friend who has a Chiari malformation where that foramen magnum is enlarged.
Mm-hmm.
He deals with severe migraines and seizures. And no, you wouldn't survive.
Thank you.
Welcome, of course. Another thing is if you think about, the lack of cerebral spinal fluid that maintains that buoyancy.
Oh yeah, you could probably start getting that hydrocephalitis.
Hydrocephaly, or you would
Yeah.
just, if you start messing with structure, you mess with function too.
All right, thank you. I'll let him know.
Of course. Yeah, of course. How old is he, Sam?
He's 10.
Okay, he's a cute kid. I can see his picture from time to time.
Oh, thank you. Have a good night. Thank you.
too.
Have a good night. Thank you.
Anything.
Okay.
Are you going to be sending email out this week of due dates?
All right, so you can do
Jen,
the rest.
say that one more time.
Are you going to be sending an email out of due dates?
Let me try tomorrow. Let me try tomorrow night.
Okay. Or has anything changed, or is it still the same this week from what was last week?
Yes.
Okay. That's okay. Then I know. I just want to make sure nothing was added or changed or.
You know what? And if I added or changed things, I would extend them out.
Okay. Cool. Got it. Thank you.
You're welcome.
Have a good night.
You too.
Hi, sorry.
Zena, stop apologizing.
Chapter 15, I see that it says like the due date was for the April 11, but whenever I tried to do it yesterday, it won't let me at all. It won't open up.
Chapter 11.
Yeah, but I can see it there. It's saying due April 11 at 11:59.
How old are your kids, Zena?
5 and 6.
Oh, they're really little, and they're right on top.
Yeah, they are. I had them back to back, so they're tiny, tiny people.
How close are they in age?
They're I think, 10 months apart. The one was
Holy
born
cow.
premature, so yeah.
All right. Yeah, they're really close in age.
Yeah,
Wow.
and they're really close, even like in person, like, they're best friends. Like you can't separate them, and
That's awesome.
yeah, they're really close friends, so it's great.
But wow, that's pretty amazing. Oh. All right, so smart book. You know what? I would call McGraw-Hill. You may have a pop-up blocker.
Oh, okay. Okay.
Try calling McGraw-Hill. If they can't help you, text me or email me back.
Okay, I'll do that, because I tried it with both of my computers. For whatever reason, it won't.
Oh,
let me
wait. No, I see the problem. Bear with me. I'm so sorry.
No, it's okay.
It's not opening until tomorrow. But there's no reason why I can't open it right now. So give me 1 second.
Okay,
I'm sorry. I'm a little brain dead at this point, and I missed that.
Oh, it's okay.
And that due date is changing, right? Because that one's not
I
due.
will push it back.
Okay.
Push it back, push it back until the 25th. I can't remember when your semester ends. But we'll push it back until the 25th, and I'm gonna open it right now. It'll open at 8:50.
Okay.
Yeah, I'm glad you didn't just call McGraw-Hill like I said, because then I would be hearing back. It's so much easier not to have to hear back, but we won't say anything as I'm still being recorded. Let's see here, chapter 15. And an assignment. Yeah, I don't know why I didn't. What I should do is just open them all up so that they open like the first day of class. Because so people like you who have a crazy schedule,
Yeah,
can just work on it when you can.
that's true. Davina, it's time to go to bed. It's late. What? No, they're not. They're a tissue. She said, are our lungs metal?